Hi, everybody. I am sitting here. I keep saying it's me, like it's going to be somebody else. But I am actually sitting in my bed because my husband's out watching TV. And I've got a bunny on the floor walking around. I've got one cat on my bed, another cat's on the other side of the room. And, life is normal, I guess. All right, so I had some suggestions that people wanted me to do the whole screen. So I'm going to do that. What a good suggestion. And then to use the little buttons. Simple but easy. All right, so we left off on this yesterday. Remember these pictures right here? This one is a rod shaped. This one is actually, this spiral word should be over here, and this spherical word should be over here. So this is like a spirocheta. Um, one of, sorry, this is spiral. Um, hmm, I can't remember. It's one of the STDs. I can't remember which one. But this one right here is intercocci. That's like E. coli. Okay. So this was yesterday. All right. This one right here. These pictures are right. Staph is usually going to be. Oops. See, you can't push the buttons. Okay. Staph is usually going to be blue, and then strep is usually going to be yellow. So you can see that there's streptococci and staphylococci. Okay? Circular cocci. All right. Oops, I keep doing the wrong button. Okay. Biologists group most species of bacteria into two different categories based on the structure of their cell walls. Remember, gram-positive has a thick layer of peptidoglycan on the top, and then gram-negative has some peptidoglycan, but then there's another covering on it, and you'll have to go back a couple of pictures. Actually, I could probably do that for you real quick, because it'll make more sense if I do that. Just can't remember exactly. Here. Okay, bear with me. Oh, here it is. All right, so this is gram-negative. So this is gram-positive. It has a huge peptidoglycan, and on this one, it has the outer membrane here, and then it has peptidoglycan right here. Okay, so gram-positive and gram-negative, and that's what we're going to be staining when they call this a gram stain. Okay, now this is something if you take Miss Schneider's microbiology class, you will actually do in class is you will gram stain things. And you can see the purple ones are going to be because of the positive ones. Wait, pink is purple. Wait. Yeah, purple is going to be gram positive, and the gram negatives are going to be pink. I'm sure. Hold on. Let me come back to that. Biologists group most species of bacteria into two categories based on the structure of their cell walls, and they examine this in a laboratory technique called gram staining. So it's based on because you have to use violet and you have to use iodine and all these different things, and so that's what, not iodine. Oh, it's called a blue color. I can't remember what it is. But this right here, you can see that there's rods here and there's cocci right here. And so these are turning. So I'm pretty sure that the purple is gram positive, and the Will, gram positive is purple. Okay, yeah, sorry. My husband, he was a lab tech. All right, so to determine, okay, so next one. Oh, there it is. Okay, so gram staining identifies bacteria, stains, stains the polymer peptidoglycan. That's what's staining it. So gram positive is going to stain purple, more peptidoglycan. So this is right here. And then gram negative is going to stain pink or red because it has less. And you can see that these are rods and these are circles. That doesn't mean that. It's just based on the most circular ones are going to be gram positive. Most rods are going to be gram negative, but that's not always the case. Gram positive, peptidoglycan. Gram positive, purple, peptidoglycan. Gram negative, less or not as much, pink. Okay? Gram positive, purple, peptidoglycan. Gram negative, not as much, pink. Okay. All right, here's a picture of it too. Here's the gram-positive with the peptidoglycan, which is all this. And then here on the gram-negative, this is peptidoglycan. It's not that much. And so that's why it's staining pink, because whatever they're using to stain, there's a bunch of different things they use. That's staining this outside. Here, it stains this. So that's why it's blue or purple, okay? Because of the violet. Okay, some gram-positive bacteria can form a thick-coated resistant structure called an endospore when environmental conditions become harsh. The endospore contains the cell's DNA and is able to survive the harsh conditions. This is good for the bacteria. This keeps them alive. So even if the harsh conditions are antibiotics or some kind of alcohol or iodine to kill germs, it can still live. But that's bad for us because it won't kill the bacteria. So endospores are good for the bacteria so they don't die, but it's bad for us if we're trying to kill them like in our body. So endospores are good for the bacteria, bad for us because it can basically keeps them safe from all the things we're trying to do to kill it. All right. Endospores can resist high temperatures, strong chemicals, radiation, drying, and other environmental extremes. When conditions return to normal, the endospore gives rise to normal bacteria cells. It's kind of like armor on one of those superheroes. I can't remember which one who wears armor. Maybe Iron Man? That makes sense. Okay, so here it is right here. And you can see that maybe there's some antibiotic and it's building this endospore all the way around. And look, there's another endospore on another one, and then it becomes free of it. So it only builds the endospore when it's under attack, basically, when there's strong chemicals, radiation, drying out, or other environmental concerns to the bacteria. And then when it's not there, sometimes it'll return back to normal. Kind of smart, right? All right. Species of the genera Bacillus and Clostridium can form endospores. Okay, so some bacilli, which are rods, can form these endospores, and Clostridium right here. Okay, the endospores C. botulium can germinate in improperly sterilized food. So botulism is a big, or used to be a big thing. It still kind of is. If food is not preserved enough in a can or in a jar, you can get botulism, and it can be really bad. And so my mom always used to look at canned goods to see if they had a bubble on the top of it, because that would mean that it had botulism in it. I don't hear about botulism as much anymore, so there's that. This right here is an endospore. I'm going to skip that. Okay. So prokaryotes can exchange pieces of DNA that can be added into the cell's DNA without replication. It's called recombination. Okay, so here it is. Here's the bacterial chromosome, okay, and here's the bacteria cell. It goes into here, and this bacteria chromosome, the genetics in it, goes into here, and it recombines into this one, okay? So this can be good, or this can be bad. So here's another picture of it. So you've got a normal cell right here, and then you've got a cell that right here that has something that the other cell doesn't. So you can see that some of this comes off and goes to the normal cell, and then it becomes part of the normal cell. So that can be like how antibiotics become resistant how bacteria becomes resistant to an antibiotic. Antibiotics used to work for everything, and now we're getting a lot of things that are resistant to antibiotics. Like a lot of sexually transmitted diseases are becoming immune to different antibiotics, and that's really bad because then we can't treat them. Okay, so here's another one. Okay, here's your donor bacterium, and this is a virus right here, and you can see that basically it's coming in here, and they're coming in and they're taking over and making the cell different. All right, same thing. All right, prokaryotes provide nutrients to humans and other animals. Prokaryotes live in digestive system of animals. They make vitamins, they break down food, and they fill niches. A niche or a niche is like a job. So some things can't do it, but they'll find a niche where they can actually work. Okay, so these can live in the digestive system of animals. Bacteria lives in our gut. So this is what they're talking about. Okay, I'll show you this tomorrow. Bye, y'all.